Good day, ma'am. So today we're going to discuss our given topic that is all about flow cytometry. What is flow cytometry? It is a technique or powerful technique used in biology and medicine to analyze and characterize cells based on various properties. It involves the use of a flow cytometer, which we're going to see later on, is a specialized instrument that passes cells in a fluid suspension through a laser beam. As cells pass through the beam, they scatter light and fluorescence emitted by fluorescently labeled molecules within the cells is detected. This process allows scientists to examine multiple characteristics of individual cells such as size, shape, granularity, and the presence of specific proteins or molecules. Flow cytometry is widely used in research, clinical diagnostics, immunology, hematology, and various other fields to study and sort different types of cells in a high throughput manner. In short to that, flow cytometry is a technique for counting, examining, and sorting microscopic objects suspended in a fluid based on their optical properties. So as we've seen in the illustration, that's what flow cytometer looks like.
And now, let's proceed to the importance of flow cytometry. Flow cytometry is incredibly important because it allows scientists and healthcare professionals to analyze and understand the characteristics of cells in a quick and accurate manner. Flow cytometry enables the identification, counting, and sorting of different cell types as well as the measurement of expression of protein expression and DNA content. This technology has widespread applications in research and clinical settings, aiding in the study of disease, immunology, cancer diagnostics, drug development, and stem cell research. Its versatility and ability to provide detailed insights into cellular properties make flow cytometry an indispensable tool by advancing our understanding our human health and disease.
Hello everyone, my name is Alvi Casisomoza and my topic will be all about the three systems of flow cytometry. Before anything else, let us define first what is flow cytometry. From the word itself, flow means fluid, cyto means cell, and metry means measurement. Obviously, flow cytometry means fluid cell measurement. Fluid cells like red blood cells and white blood cells. Traditional flow cytometers consist of three systems, the fluidics, optics, and electronics. Let us talk about fluidics first. So the main function of fluidic system is to transport particles in a fluid stream to the laser beam for interrogation point. So when we say interrogation point, this is the place within the flow cytometer where the laser light hits the individual particles as they pass in front of the laser, one at a time. So basically, the fluidic system consists of sheath fluid, usually a buffered saline solution that is pressurized to deliver and focus the sample to the laser intercept or interrogation point where the sample is analyzed. Now let us talk about the second system of flow cytometer. So the optical system. Now, optical system consists of excitation optics and collection optics. So the excitation optics, it is where laser beams are directed towards the flow cell for interrogation of the sample core. Now, excitation components include the laser beam, shaping prism, and lenses to route the laser beams to the flow cell. Now, the collection point. The collection point involves the gathering of scattered and emitted light from the sample and the routing of collected light to the appropriate detectors. So it is clearly understandable that the collection point gathers the data of scattered and emitted light from the sample. Now lastly, let us talk about the final component of a flow cytometer. Now, let's talk about the electronics system. So in a flow cytometer, electronic system is responsible for digitizing and processing the photocurrent from the detector and saving it for subsequent analysis. So during this stage, electronic system is going to analyze data, gather data.
Good day, everyone. I'm here to discuss the process of flow cytometry. So, flow cytometry is used to count and analyze the size, shape, and properties of an individual cells within the heterogeneous populations of cells. So, how does the flow cytometry analyze the size, shape, and properties of an individual cell? A flow cytometry contains several key components, including the samples, the fluid that moves the sample into the flow cytometry, the lasers, the optics that gather the light, the detector that sends the light, and the computer system to output the data into the form that can be analyzed by the researcher. So once the sample is placed into the flow cytometry, the sample will suck, the machine will suck up the samples and mix the sample into the saline solution in the cytometer. Then it leads to the cell suspension through the narrow channel, causing the cells to form a single fine line before the cells pass through the laser at the interrogation point. So each cells within the samples then moves across the laser beam, allowing each cells within the samples to be individually analyzed. Each cells passes through the laser beam. The laser beam will scattered into multiple directions. The flow cytometry detect this light scattered in a form of forward manners called the forward scattered, and scattered the light scattered on the sideways manner called the side scattered. So the amount of forward scattered light for each cells is detected by the detector on the far side of the cells from the laser. So forward scattered is proportional to the size of the cells. The detector convert the scattered light into the voltage pulse with a directly proportional to the amount of forward scattered light. So the computer converts this data into the histogram plot with the amount of forward scattered light and the x-axis and the amount or the number of cells in the y-axis. So the amount of the side scattered light is detected by the detector located perpendicular path of the laser beam side of the laser beam. The side scattered proportional to the shape and internal complexity of a cell. So just like the forward scattered light, the flow cytometry convert the detected side scattered light into the voltage pulse, which is directly proportional to the amount of side scattered light. So by analyzing the forward and side scattered light data together, the researcher can understand the size, shape, and complexity of a cell. In addition, the analysis of the forward and side scattered light they can be allowed to the researcher to divide the heterogeneous population of cells into the individual populations with the varying size, shape, and complexity.